Good morning, students. We will continue the topic carbohydrate metabolism. Today's competency is competency number 3.4 and 3.5. In these two competencies, we are studying various pathways of carbohydrate metabolism. Before going to the pathway, I will summarize what is metabolism. Metabolism is combination of catabolism and anabolism. Catabolism means breakdown, anabolism means synthesis. In our body, the compounds are continuously synthesized and broken down. So that combination of these two is called as metabolism. If we analyze the elements present in our compounds, then we will found that the major elements found in these compounds are carbon, hydrogen and oxygen, along with some minor elements like nitrogen. Therefore, during synthesis, the main reactions are reduction and carboxylation. Reduction adds hydrogen atom, whereas carboxylation adds carbon and oxygen to form the various compounds. The enzyme required in the reaction of reduction are known as reductase enzyme, whereas the enzyme required for carboxylation reaction are carboxylase enzymes. Similarly, catabolism is nothing but the breakdown of the compound which is formed during anabolism. So it is done by again two process, oxidation and decarboxylation. Oxidation removes two hydrogen atoms from the substrate and decarboxylation removes the carbon dioxide from the substrate. For the process of oxidation, the enzyme required is generally dehydrogenases, and decarboxylation reaction requires enzyme decarboxylases. So this is in brief about the summary of metabolism. It is not the metabolism of carbohydrate, it is the metabolism of all the compounds in the body. Now we will start the first pathway of carbohydrate metabolism, that is glycolysis pathway. It is also called as Embeden-Meyerhof pathway. First, the definition: breakdown of glucose into pyruvate in the presence of oxygen, or breakdown of glucose into lactate in the absence of oxygen is called as glycolysis. Site means the place where this pathway occurs. This pathway occurs in the cytoplasm of all cells. All of you have studied this pathway in 12th standard, so there is no need to go in details of this pathway. So in the first step, glucose is converted into glucose 6-phosphate with the help of enzyme hexokinase. Here glucose is phosphorylated with the help of inorganic phosphate donated by ATP, and ATP is converted into ADP. In the second step, glucose 6-phosphate is converted into fructose 6-phosphate, which are isomers of each other. So enzyme is isomerase. In the third step, fructose 6-phosphate is again phosphorylated for the second time to form fructose 1,6-bisphosphate. Again the inorganic phosphate is provided by ATP, and the enzyme required here is phosphofructokinase. So you have seen two ATPs are utilized for the phosphorylation of the substrate at various steps. Now this fructose 1,6-bisphosphate is then broken down into 3 carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, with the help of enzyme aldolase. Now the dihydroxyacetone phosphate is later on converted into glyceraldehyde 3-phosphate with the help of enzyme isomerase. because they are isomers of each other. So ultimately here we have got two molecules of glyceraldehyde 3-phosphate from one glucose molecule. Now in this fifth step, two molecules of glyceraldehyde 3-phosphate are converted into 1,3-bisphosphoglycerate. Again here phosphorylation takes place, but here phosphorylation is not through ATP, it is by addition of inorganic phosphate, direct addition of inorganic phosphate. At the same time, here the first oxidation occurs. That means two hydrogen atoms are removed from glyceraldehyde 3-phosphate, which are accepted by NAD and get reduced to NADH. So two NADH molecules are synthesized as two glyceraldehyde 3-phosphate molecules are converted into two molecules of 1,3-bisphosphoglycerate kinase. The enzyme required for this step is dehydrogenases. The complete name is glyceraldehyde 3-phosphate dehydrogenase. Later on, this 1,3-bisphosphoglycerate undergoes dephosphorylation. One phosphate group is removed from the first position and it is added to ADP to synthesize ATP. So here ATP molecules are synthesized. This type of synthesis of ATP is called as substrate level phosphorylation. That means synthesis of ATP by adding a phosphate directly from the substrate is called as substrate level phosphorylation. In general, ATP synthesis occurs in the electron transport chain by chemiosmotic theory. But here the ATPs are directly synthesized by taking an inorganic phosphate from the substrate. So this type of ATP synthesis is called as substrate level phosphorylation. The enzyme required here is kinase. The complete name is 1,3-bisphosphoglycerate kinase. Now the 3-phosphoglycerate kinase is converted into 2-phosphoglycerate with the help of enzyme mutase. Here the phosphate group from the third carbon is shifted to second carbon. Now 2-phosphoglycerate is later on converted into phosphoenol pyruvate with the help of enzyme enolase, and this phosphoenol pyruvate undergoes dephosphorylation by removing the inorganic phosphate. Again this inorganic phosphate is directly added to ADP to form ATP. So this is also an example of substrate level phosphorylation, and ultimately pyruvate is formed. The enzyme required for this step is pyruvate kinase. If oxygen is present, this is the end product of glycolysis. But if oxygen is absent in the cell, then the pyruvate is reduced to lactate. Two hydrogen atoms from a coenzyme NADH are added to pyruvate to form a lactate with the help of enzyme lactate dehydrogenase. So when oxygen is present, then the end product is pyruvate, and when oxygen is absent, the end product is lactate. When the pyruvate is converted into lactate, the NADH is utilized, and that NADH is synthesized in the fifth step of glycolysis, where the dehydrogenase will convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. So that NADH which is formed in the fifth step of glycolysis is used here for reduction of pyruvate to lactate. So this is the complete pathway of glycolysis. Now come to energetics of glycolysis. As there are two types of glycolysis, aerobic glycolysis means in the presence of oxygen, and anaerobic glycolysis means in the absence of oxygen. So first we will see the energetics of aerobic glycolysis. On the left side I have written the enzyme, on the right side I have written the ATP calculation. So in the first step of glycolysis, hexokinase utilizes one ATP molecule for the phosphorylation of glucose to glucose 6-phosphate. So one ATP is utilized. Then in the third step of glycolysis, the enzyme phosphofructokinase again utilizes one ATP molecule for the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. So again one more ATP is utilized. In the fifth step of glycolysis, glyceraldehyde 3-phosphate dehydrogenase generates 2 NADH molecule from 2 molecules of glyceraldehyde 3-phosphate, because one glucose molecule will give 2 molecules of glyceraldehyde 3-phosphate. So 2 NADH molecules are regenerated. Each NADH when enters into the TCA cycle gives 2.5 ATPs. So 2 NADH will give 5 ATPs. Then the sixth step, the enzyme is 1,3-bisphosphoglycerate kinase. Here this enzyme causes dephosphorylation of the substrate, and that phosphate is added to ADP to synthesize ATP. So 2 ATPs are synthesized in this step. Then the enzyme pyruvate kinase again causes dephosphorylation of the substrate phosphoenol pyruvate, and that inorganic phosphate is used for the synthesis of ADP to ATP. So in this way, if you calculate, total 9 ATPs are formed. 5 ATPs are obtained from NADH molecules and 4 ATPs are obtained from substrate level phosphorylation, out of which 2 ATPs are utilized for the initial phosphorylation of the substrate by enzyme hexokinase and phosphofructokinase. So the net gain of 7 ATPs takes place in aerobic glycolysis. Anaerobic glycolysis is same except the fifth step, where NADH will give 5 ATPs in aerobic glycolysis. But in case of anaerobic glycolysis, we have seen the NADH molecule which is synthesized in the fifth step is by dehydrogenase is utilized by the enzyme LDH for conversion of pyruvate to lactate. So the 5 ATP molecules of NADH are lacking here. The remaining are same. So here 4 ATP molecules are synthesized by substrate level phosphorylation, out of which 2 are utilized for phosphorylation of the substrate by the enzyme hexokinase and phosphofructokinase. So net gain of 2 ATPs occurs in anaerobic glycolysis. Now come to regulation of glycolysis. Glycolysis is regulated by allosteric mechanism. The enzymes of glycolysis like hexokinase, phosphofructokinase and pyruvate kinase, these enzymes are allosteric enzymes. Allosteric enzyme means the enzymes which has 2 different sites. One site is called as active site for binding of substrate, and another site is called as allosteric site for the binding of modifier. Modifier may be stimulator or activator. If the modifier is stimulator, then that stimulator binds at the allosteric site of the enzyme. Due to this binding, the affinity between the active site and the substrate increases and the reaction rate increases. If the modifier is inhibitor, Then that inhibitor binds at the allosteric site of the enzyme and it brings the conformational change in the shape of active site so that substrate will not fit into the active site of the enzyme. Therefore, the rate of reaction decreases. So, the cell will synthesize either stimulator or inhibitor according to the need of the pathway. So, this slide shows the allosteric stimulators and allosteric inhibitors of glycolysis pathway. The main aim of glycolysis is it is a preparatory stage before citric acid cycle or TCA cycle, where the glucose is completely oxidized and ATPs are synthesized. So, generally the rate of glycolysis increases when cell requires energy and the rate of glycolysis decreases when the enough ATPs are present inside the cell. So, the first allosteric enzyme of glycolysis is hexokinase. It is inhibited allosterically by glucose 6-phosphate. Glucose 6-phosphate is the product of the enzyme hexokinase. Hexokinase will convert glucose into glucose 6-phosphate. So, it is a product. Generally, the products of the enzyme acts as a inhibitor and the substrate of the enzyme act as a stimulator. So, see this here, the substrate, suppose substrate is A, is converted into B with the help of enzyme E. If the concentration of A increases, which is a substrate, then it stimulates the enzyme E. But if the concentration of B, that is product, increases, then it inhibits the enzyme E. Generally, this is the mechanism for regulation of various enzymes. So, here glucose 6-phosphate is the product of hexokinase. So, it acts as a inhibitor. Now, come to the next enzyme, that is phosphofructokinase. The stimulator for phosphofructokinase are, first is ATP. When the concentration of ATP inside the cell increases, the cell will slow down the glycolysis pathway. So, ATP itself acts as a allosteric inhibitor. The second inhibitor of phosphofructokinase is citric acid or citrate. Citrate is formed in the TCA cycle. When the ATP requirement of the cell is low, at that time the TCA cycle operates in slow space or slow speed. Therefore, citric acid get accumulated inside the mitochondria. When saturation occurs, then that citric acid enters into the cytoplasm and it will inhibit the enzyme phosphofructokinase. Then, when these various acids like citric acid, pyruvic acid, lactic acid inside the cytoplasm increases, the pH of the cytoplasmic compartment of the cell decreases. They inhibit the enzyme phosphofructokinase. And the next inhibitor is cyclic AMP. Cyclic AMP is generally formed from ATP with the help of enzyme adenine cyclase. This adenine cyclase is activated when the concentration of ATP is high inside the cell. So, formation of cyclic AMP itself indicates that the ATP concentration of cell is high, and the cell does not want more ATPs. So it tries to inhibit the pathway of glycolysis. Now come to allosteric stimulators. AMP, which is formed by the hydrolysis of ATP, act as a stimulator. Presence of AMP inside the cytoplasm indicates that the concentration of ATP decreases. So AMP stimulate the glycolysis pathway to regenerate the ATPs. Fructose 6-phosphate is a substrate of the enzyme phosphofructokinase. When the concentration of fructose 6-phosphate increases, as I have told you, the substrate always stimulates the enzyme. So fructose 6-phosphate stimulates the enzyme phosphofructokinase. The other stimulators are phosphofructokinase 2 and fructose 2,6-bisphosphate. Which are these compounds? We will see. When the concentration of fructose 6-phosphate increases in the cell, then that fructose 6-phosphate activates the enzyme phosphofructokinase 2, which is not an enzyme of glycolysis. Phosphofructokinase is an enzyme of glycolysis, but phosphofructokinase is not an enzyme of glycolysis. It is activated only when fructose 6-phosphate concentration is high. Now this phosphofructokinase 2 then convert the fructose 6-phosphate into fructose 2,6-bisphosphate with the help of ATP. Now this fructose 2,6-bisphosphate act as a positive regulator or positive stimulator of the enzyme phosphofructokinase. So in this case, all three, fructose, phosphofructokinase 2 and fructose 2,6-bisphosphate, act as a allosteric stimulator of the enzyme phosphofructokinase. Now come to the next enzyme, that is pyruvate kinase. Inhibitors are same, ATP and cyclic AMP. When the concentration of ATP is high, the pathway of glycolysis is inhibited, and the stimulator is fructose 1,6-bisphosphate, which is a not a substrate, but it is a pro-substrate of this enzyme pyruvate kinase. So in this way, the glycolysis is regulated by allosteric mechanism. Now come to significance of glycolysis. It is the only pathway that occurs in all cells of the body. Second, it is the pathway which provides energy for erythrocyte. All of us know that erythrocyte are lacking in mitochondria, endoplasmic reticulum, Golgi apparatus. They does not have anything. They have just cytoplasm. So glycolysis is the only pathway that occurs in RBCs. TCA cycle, electron transport chain, these pathways occurs in mitochondria, but in RBCs mitochondria is absent. So glycolysis is the only pathway which provide energy to erythrocyte. During heavy exercise or in any conditions of hypoxia, due to deficiency of oxygen, the TCA cycle and electron transport chain stops. At that point, the requirement of energy is fulfilled by the glycolysis pathway, because glycolysis can occur both in the presence of oxygen as well as in the absence of oxygen. So in case of hypoxia, the main pathways like TCA cycle and electron transport chain stops. So energy input from this pathway stop, but the tissue requires energy, so it is supplied by glycolysis. Next significance is, as I have already told you, that glycolysis is a preliminary step before complete oxidation of glucose. Complete oxidation of glucose means conversion of glucose into carbon dioxide and water molecule. That takes place in TCA cycle. Glycolytic pathway also provides carbon skeletal for synthesis of non-essential amino acids. Like pyruvate is converted into alanine with the help of enzyme alanine transaminase. Similarly, the dihydroxyacetone phosphate, which is formed in the glycolysis as an intermediate of glycolysis, can be converted into glycerol, and that glycerol is further esterified to form triacyl glycerol, which is a fat. Then the last significance is majority of the reactions of glycolysis are reversible. The only 3 reactions of glycolysis are irreversible, whereas remaining reactions are reversible. The 3 irreversible reactions are: first, the reaction catalyzed by the enzyme hexokinase; second, the reaction catalyzed by the enzyme phosphofructokinase; and the third is the reaction catalyzed by the enzyme pyruvate kinase. So these 3 are irreversible reactions, and the remaining reactions of glycolysis are reversible. Therefore, during the process of gluconeogenesis, where pyruvate is converted into glucose, during that pathway, the reversible reactions of glycolysis is used for the synthesis of the glucose molecule. Next cycle is Rapaport Leubering cycle, which is generally studied along with glycolysis. So first I will show you what is Rapaport Leubering cycle. So this Rapaport Leubering cycle occurs in RBCs. If you see the slide, I have shown the glycolysis pathway in a straight chain, but at one step 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate, and then that 2,3-bisphosphoglycerate is converted into 3-phosphoglycerate. That is a diversion of one step of glycolysis. In glycolysis in other tissues, 1,3-bisphosphoglycerate is directly converted into 3-phosphoglycerate, but in RBCs 1,3-bisphosphoglycerate is first converted into 2,3-bisphosphoglycerate with the help of enzyme bisphosphoglycerate mutase. Here there is a shifting of phosphate from first carbon to second carbon, so mutation takes place, so enzyme is mutase. Later on, that 2,3-bisphosphoglycerate is dephosphorylated to form 3-phosphoglycerate by the enzyme 2,3-bisphosphoglycerate phosphatase. So in RBCs, the glycolysis occurs by the diversion, and the diversion of only one step. Remaining steps are same like the glycolysis in other tissues. Therefore, Rapaport Leubering cycle is defined as it is a diversion of one step of glycolysis in which 2,3-bisphosphoglycerate is synthesized. It occurs in RBCs. The difference between the glycolysis of other tissues and RBC is that 2,3-BPG is formed in RBCs, and the second difference is, if you see, there is a substrate level phosphorylation in glycolysis, when it is converted into 1,3-bis phosphoglycerate to 3-phosphoglycerate. That substrate level phosphorylation does not occurs due to this diversion or Rapaport Leubering cycle. Here ATP synthesis does not takes place. So these are the two differences in glycolysis of RBCs and glycolysis of other tissues. Significance of Rapaport Leubering cycle, as I have told you, 2,3-bis phosphoglycerate BPG forms in the Rapaport Leubering cycle. This 2,3-BPG binds with the beta chain of hemoglobin. Each hemoglobin has 4 globin chains, 2 are alpha and 2 are beta. Bisphosphoglycerate, 2,3-bis phosphoglycerate binds with the beta chain of hemoglobin, and they decreases the affinity between hemoglobin molecule and oxygen, as a result of which hemoglobin can easily deliver the oxygen to the tissue. If 2,3-BPG is absent, then hemoglobin molecule binds with oxygen more tightly, hence it is difficult to release the oxygen to the tissue. Therefore 2,3-BPG helps the hemoglobin molecule to release the oxygen to the tissue. Under the condition of hypoxia, means oxygen deficiency, the level of 2,3-BPG inside the cell increases. More and more 2,3-BPG is formed inside the RBCs, and that 2,3-BPG binds with the hemoglobin molecule and forces the hemoglobin molecule to release more and more oxygen to the tissue, because in case of hypoxia there is oxygen deficiency to the tissue. So in this way 2,3-BPG tries to overcome the hypoxic conditions in the tissue. Level of 2,3-BPG is also increases at high altitude. Again the reason is same. At high altitude the oxygen partial pressure of oxygen in the air is low, due to which again there is hypoxia, which will lead to synthesis of 2,3-BPG inside the RBCs. The fourth significance is, as I have already told you, there is no synthesis of ATP or phosphate level phosphorylation, substrate level phosphorylation does not occurs due to Rapaport Leubering cycle. Therefore ATP is not synthesized. This is related to Rapaport Leubering cycle or clinical significance of Rapaport Leubering cycle. If hexokinase is deficient, if the enzyme hexokinase is deficient in some person, then the level of 2,3-BPG is low. Hence the affinity between hemoglobin and oxygen increases. Hemoglobin releases less oxygen to the tissue. Hexokinase converts glucose into glucose 6-phosphate. If this reaction does not take place or it occurs at slow space, then there is less concentration of 1,3-bis phosphoglycerate. Therefore the diversion, that is Rapaport Leubering cycle, also occurring at slow space. So 2,3-BPG level is also less. But if the enzyme pyruvate kinase is deficient, then the level of 2,3-BPG inside the RBCs is high. Why? Pyruvate kinase catalyzes the conversion of phosphoenol pyruvate to pyruvate. If this enzyme is deficient, then phosphoenol pyruvate get accumulated, due to which all the substrate like 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bis phosphoglycerate and glyceraldehyde 3-phosphate get accumulated. Therefore, the concentration of 1,3-bis phosphoglycerate increases. When the concentration of 1,3-bis phosphoglycerate increases, it is diverted to form 2,3-bis phosphoglycerate. Therefore, in case of pyruvate kinase deficiency, the level of 2,3-BPG is high, whereas in case of hexokinase deficiency, the level of 2,3-BPG is low. Now come to the clinical disorders related to glycolysis. The most common is lactic acidosis, means accumulation of lactic acid. It is generally seen in exercise. The reason is oxygen deficiency. Hypoxia means again same oxygen deficiency. In shock, there is circulatory failure. So again, the tissues will get less oxygen. In case of pulmonary failure, there is difficulty in gaseous exchange. So again there is hypoxia or oxygen deficiency. Lactic acidosis is also seen in alcoholic person. This is because when ethanol is metabolized, it is converted into acetaldehyde by the enzyme alcohol dehydrogenase. Acetaldehyde is converted into acetic acid by the enzyme aldehyde dehydrogenase. During this process, NAD is converted into NADH. Means when one molecule of ethanol is metabolized, 2 molecules of NAD are converted into NADH. Therefore, the concentration of NAD inside the cell decreases and concentration of NADH inside the cell increases. Here you have to remember the ethanol is metabolized in the cytoplasmic compartment of the cell. Glycolysis is also taking place in the cytoplasmic compartment of the cell. So in that cytoplasmic compartment of the cell, there is deficiency of NAD because NAD is reduced to NADH. NADH concentration increases, but there is deficiency of NAD. Therefore, the cell tries to regenerate NAD from NADH. And how it regenerate? By converting the pyruvate into lactate. This is the actual side. The cell tries to regenerate NAD by converting pyruvate into lactate with the help of enzyme LDH. So lactate formation increases because pyruvate is diverted for lactate synthesis. The aim of this step is to regenerate NAD. The next cause of lactic acidosis is diabetes mellitus. Normally, the pyruvate which is formed from glucose is converted into acetyl CoA with the help of enzyme pyruvate dehydrogenase. Insulin acts as a stimulator of this enzyme, or insulin stimulates the enzyme PDH. In diabetes mellitus, as there is deficiency of insulin, therefore this PDH is not completely activated. Hence, the conversion of pyruvate to acetyl CoA is taking place at slow speed. Therefore, the pyruvate is converted into lactic acid. Second cause is oral hypoglycemic drugs which are used by the diabetic patients like metformin and sulfonylureas. These drugs also causes lactic acid accumulation in the tissue. So these are the various causes of lactic acidosis. Now the next clinical disease is deficiency of glycolytic enzymes. Generally, the deficiency of pyruvate kinase, hexokinase, aldolase and phosphofructokinase is seen. If the enzymes of glycolysis are deficient, then the person presents with hemolytic anemia. Why hemolytic anemia? Because I have already told you, glycolysis is the only pathway that provide energy for RBCs. If the enzymes of glycolysis are absent or deficient, then RBCs will not get enough energy, so they become fragile and easily hemolyze. So the patient is presented with hemolytic anemia. If the hexokinase deficiency is seen, just now we have studied, then the level of 2,3-BPG is low. If the deficiency of pyruvate kinase is seen, then the level of 2,3-BPG is high. In case of phosphofructokinase deficiency, the persons are generally presented with muscle weakness. So this is about glycolysis along with Rapaport Leubering cycle, energetics, regulation and clinical diseases and disorders related to glycolysis and Rapaport Leubering cycle. Thank you very much.